Hello all. In this video we'll be talking about H2 receptor antagonist and proton pump inhibitors. First we'll begin with H2 receptor antagonist. Three drugs are given in your syllabus: Cimetidine, Ranitidine and Famotidine. Before moving to the actual H2 receptor antagonist, we'll just have a brief account of mechanism involved in the gastric acid secretion. As you know that acid is secreted from the parietal cells in the gastrointestinal tract. This picture is taken from the Goodman and Gilman's The Pharmacological Basis of Therapeutics. In the figure you can see the big square that depicts the parietal cells where the acid is formed and secreted out. And just before that you can see a rectangle that denotes superficial epithelial cells that is concerned with the secretion of mucus. And then there is the endochromaffin-like cells that is concerned with the secretion of histamine. Gastric acid secretion is a complex and a continuous process, and number of systems are involved in it, and there are both central and peripheral factors that controls the acid secretion. Three important systems are involved in the gastric acid secretion. One is the neuronal or central nervous system that acts through the acetylcholine. Acetylcholine acts through the muscarinic receptors present in the parietal cells and the ECL cells. And then the second one is a paracrine system that is through the histamine. Histamine released acts through H2 receptors in the basolateral membrane of the parietal cells and increases the acid secretion. And the third one is the endocrine system that acts through gastrin. And the gastrin released acts through cholecystokinin receptors or CCK2 receptors, and that is also involves in the acid secretion from the parietal cells. Acetylcholine or the parasympathetic system directly and indirectly controls the gastric acid secretion. As you all know that whenever you think of food, whenever you see food, or whenever you have a smell of food, you feel hungry, and that is because of the increased acid secretion in the gastrointestinal tract. And the increased gastric secretion is because of the involvement of parasympathetic system, that is the acetylcholine released from the parasympathetic system. So this released acetylcholine can directly act on the muscarinic receptors present in the parietal cells and can cause a release of gastric acid or hydrochloric acid secretion. Indirect mechanism of action of acetylcholine is that acetylcholine increases the release of histamine from endochromaffin-like cells. Endochromaffin-like cells are present in the fundus of the stomach. Acetylcholine also increases the release of gastrin from the G cells in the gastric antrum. This released histamine and gastrin are potent inducers of acid secretion. The next system that is concerned with the acid regulation is the paracrine system, that is through the action of histamine. Histamine is synthesized and released from the endochromaffin-like cells present in the fundus of the stomach. Histamine is released in response to 1. Stimulation by gastrin on CCK2 receptors, and 2. By the action of acetylcholine on the muscarinic receptors, M1 receptors present in the endochromaffin-like cells. And this released histamine stimulates the H2 receptors present in the parietal cells, and that causes the increased secretion of acid from the parietal cells. Then the third system that controls the acid secretion is the endocrine system or the gastrin released from the gastric G cells of the gastric antrum. And this gastrin is a potent inducer of gastric acid secretion. They act on the CCK2 receptors present on the parietal cells and as well as on the endochromaffin-like cells. When they act on the endochromaffin-like cells, they increase the release of histamine, and histamine in turn causes the increase in secretion of acid. So in short, you can say that there are three systems that regulates the acid secretion in the gastrointestinal tract. One is the acetylcholine. Acetylcholine is released in response to sight, smell, taste, or anticipation of food. They act on the muscarinic receptors present in the parietal cells, as well as they cause an increase in the secretion of histamine from endocrine-like cells, as well as the gastrin. Then the other two systems are one is the paracrine system through histamine, and the other one is the endocrine system through the gastrin. And this gastrin and histamine are the potent inducers of acid secretion. The acid is formed in the parietal cells, and they are secreted out from this parietal cells against the concentration gradient. It is an active process, and that secretion or the pumping is done by a pump known as proton pump or H plus K plus ATPase pump. That is, it requires for the secretion or the pumping of acid, it requires energy, so that is, it is connected with the ATPase. So energy is spent out for the secretion of acid across the concentration gradient. And for that pumping, the parietal cells have proton pump or H plus K plus ATPase. It is a pump that pushes out the acid from the parietal cells into the lumen. So till now we have seen the various factors that causes the increased secretion of acid inside the gastrointestinal tract. Now we will see there are certain factors that inhibits the acid secretion in the gastrointestinal tract, and one such inhibitor is the somatostatin. They inhibit gastric secretion. Whenever the pH of the gastric content is less than 3, the somatostatin is released, which inhibits the acid secretion by a negative feedback loop mechanism. The next mechanism that inhibits the acid secretion is through the prostaglandins. Prostaglandin E2 and I2, they are potent inhibitors of the gastric acid secretion. They play two roles. One is that they inhibit the acid secretion, and the second one is that they act on the prostaglandin receptors present in the superficial epithelial cells, and these epithelial cells causes the increased secretion of mucus as well as the bicarbonate. The released bicarbonate neutralizes the acid in the mucosal cells, thereby raising the pH, and it offers a protection against the acid mediated damage. So that's why drugs like non-steroidal anti-inflammatory drugs, they inhibit the prostaglandin synthesis in the epithelial cells, and as a result decreased mucus protection and decreased bicarbonate production, and that can cause gastrointestinal tract bleeding, ulceration, etc. Now we'll move on to the H2 receptor antagonist. So H2 receptor antagonists are those agents that reversibly blocks the H2 receptors. So what happens when H2 receptor is blocked? That causes decreased production of acid. So H2 receptor antagonist inhibits acid production by reversibly competing with histamine for binding to H2 receptors present in the parietal cells. Now we'll see what are the important structural features of H2 receptor antagonist. H2 receptor antagonists were invented as a result of intentional modification of the structure of histamine. So we have learned about H2 receptor antagonists. There were certain important structural features for H1 receptor antagonist. H2 receptor antagonists are entirely different from H1 receptor antagonist. First thing is that in H2 receptor antagonist, they are having smaller aromatic ring system. So like histamine is having imidazole ring. So H2 receptor antagonist is also having small 5-membered ring system, imidazoles, furan, pyran, etc. And for competitive antagonism at H2 receptors, imidazole ring of the histamine is not essential. Instead of the imidazole ring, furan, thiophene, 1,3-thiazole, etc. can also be used for bringing about the antagonism action. And then introduction of a methyl group at the 5th position of the imidazole ring. That causes selectivity towards H2 receptor. Histamine has a imidazole nucleus, and that histamine acts with equal affinity at both the H1 and H2 receptors. So introduction of a methyl group at the fifth position of the imidazole ring makes that molecule more selective towards the H2 receptors rather than at the H1 receptors. Now, like H1 receptor antagonist, H2 receptor antagonists also have a terminal nitrogen atom, and the distance between, or the number of atoms between the aromatic ring system and the terminal nitrogen atom is usually 4. That is, the aromatic ring system and the terminal nitrogen atom is separated with the equivalent of 4 carbon atoms, and that 4 carbon atoms appears to be necessary for the optimum antagonistic activity. And the instead of carbon atom, the carbon atom can be replaced by isosteric thioether linkage. Now we will see an example for an H2 receptor antagonist. What you see here is a structure of a commonly used H2 receptor antagonist, ranitidine. You can see an aromatic ring system separated from the terminal nitrogen atom by an equivalent of 4 carbon atoms. And then the aromatic ring system is a smaller aromatic ring. It is not imidazole, it is furan. Then at the terminal nitrogen atom is a guanidine, substituted guanidine. So that explains all the structural features of H2 receptor antagonist. Now we'll see the first H2 receptor antagonist, Symmetidine. You can see that Symmetidine is a 5-methyl imidazole derivative. The terminal nitrogen atom is a part of the guanidine group, substituted guanidine group. The 2 nitrogen atom of the guanidine group is substituted with a cyano group and a methyl, N-methyl group. So N-cyano, N-methyl guanidine, and this guanidine is necessary for the antagonistic property. Then the terminal nitrogen atom and the 5-methyl imidazole ring is separated by a equivalent of 4 carbon atom. You can see a methyl ethyl thioether chain. A sulfur atom is there in the chain that separates the aromatic ring system and the substituted guanidine group. Now, Cimetidine is the first clinically effective and relatively safe full H2 receptor antagonist. It is a short acting and hence requires frequent dosing. An adverse effect of Cimetidine is that its action on the hormones. It was found to have anti-androgenic activity, and that can lead to gynecomastia, confusional states in elderly and in those with decreased renal function. Another problem with Cimetidine is that it inhibits cytochrome P450 enzyme system and thereby inhibits metabolism and elimination of other drugs metabolized by these enzymes. Now coming to the next H2 receptor antagonist, Ranitidine. It is one of the most widely used H2 receptor antagonist. You can see the structure of Ranitidine on the screen. It is an aminoalkyl furan derivative. Unlike histamine and Cimetidine, which contains an imidazole ring, Ranitidine carries a aminoalkyl furan derivative. And the terminal nitrogen atom, you can see a unsaturated carbon atom, ethene carbon atom. And 1 carbon atom of that ethene group is substituted with a nitro group, and the other carbon atom is substituted with a diamine group, and one of the nitrogen atom carries a methyl group. So N-methyl 2-nitro-1,1-ethene diamine group forms the terminal nitrogen atom of the ranitidine. And then the linkage is a thioether, methyl-ethyl thioether chain connects the aromatic ring with the terminal nitrogen atom. It is more potent than Cimetidine, but it is less potent compared to Famotidine, the next that you will be studying. And then they are weak inhibitors of cytochrome P450 enzyme system, and therefore it does not have much drug interactions. Next, we will see the most potent H2 receptor antagonist, that is Famotidine. Famotidine, the aromatic ring system present in Famotidine is a thiazole ring, thiazole derivative, and you can see a guanidine substituent in the thiazole ring. You can consider this famotidine as a thiazole isoster of Cimetidine, and it contains a guanidine substituent in the thiazole ring that mimics the imidazole ring of Symmetidine. The terminal nitrogen atom forms a part of the guanidine group. It is substituted with a sulfonamide group. Sulfonamidoguanidine forms the terminal nitrogen atom. Then famotidine is a highly potent H2 receptor antagonist. It does not have any anti-androgenic activity and hence does not cause gynecomastia. It also does not inhibit cytochrome P450 enzyme system and hence does not cause any drug interaction. Coming to the uses of H2 receptor antagonist, they inhibit both basal and nocturnal gastric secretion. Nocturnal means during the night time. They also inhibit secretion stimulated by food and pentagastrin. They are useful for the short-term treatment and management of duodenal and benign gastric ulcers. Then diseases due to hypersecretion of acids, esophagitis, heartburn, gastroesophageal reflux diseases, etc. Now coming to the next class of drugs that interfere with the acid production is the proton pump inhibitors or gastric H plus K plus ATPase inhibitors. We will come back to the figure that we have seen during the regulation of acid production in the gastrointestinal tract. We have seen that there are three systems that regulate the acid production and secretion in the gastrointestinal tract. So whatever be the mechanism by which the acid secretion is increased, ultimately the acid formed in the parietal cells has to be pumped out against the concentration gradient by using this proton pump. And this proton pump inhibitors are the most effective suppressors of gastric acid secretion. They inhibit both basal and stimulated acid secretion. They are most effective for the anti-ulcer therapy. Their action is independent of secretagogues like histamine, gastrin, acetylcholine, etc. Examples of proton pump inhibitors are omeprazole, lansoprazole, rabeprazole, pantoprazole, etc. And you need to study only two drugs, omeprazole and pantoprazole. Coming to the chemistry of proton pump inhibitors, this is the general structure of proton pump inhibitors. You can see a pyridine ring and then also you can see a benzimidazole ring on the right side. And this benzimidazole ring and the pyridine ring are connected by a sulfonyl group. And so you can think of this proton pump inhibitors as a substituted pyridine derivative with a benzimidazole ring connected to it by a sulfonyl group. These proton pump inhibitors are prodrugs and they require activation in the acidic environment. So once it reaches the blood, it enters the parietal cells from the blood, and they are basic in nature. These compounds are basic in nature and hence they accumulate in the acidic secretory canaliculi of the parietal cells. And there they are activated by a proton catalyzed process, that is in presence of the acid medium, H plus medium, they get activated, and that results in the formation of a thiophilic sulfonamide or sulfenic acids, which again form a cyclic amide with the nitrogen of the benzimidazole ring. So what you see on the screen is the representation of mechanism of action of proton pump inhibitors. Proton pump inhibitors are benzimidazole derivative, and so these benzimidazole derivative in acid medium, in presence of acids H plus, they undergo a proton catalyzed reaction to form a sulfenic acid. Sulfenic acid is SOH, and that sulfenic acid again undergoes a proton catalyzed reaction to form a sulfenamide, and this sulfonamide reacts with the enzyme cysteine of the proton pump ATPase enzyme, and it forms a covalent complex with the enzyme. The end result is that the proton pump is inhibited. Now we will see the structural feature of the two classes of drug, omeprazole and pantoprazole. We have seen the general structure of proton pump inhibitors, and these are the different substitutions present in the omeprazole and pantoprazole. Then coming to the uses of proton pump inhibitors. These proton pump inhibitors inhibits both basal and stimulated acid secretion, and they are most effective anti-ulcer therapy, and their action is independent of secretagogues like histamine, acetylcholine, etc. And they are used for the treatment of gastric duodenal ulcers, erosive esophagitis, then GERD, duodenal ulcers associated with H. pylori along with antibiotics, etc. So that's all. Thank you very much.